Today we present to you DeMol, and we are a group of final year pharmacy students who believe that the future of pharma is autonomous, intelligent, and open. So, for the past few years, we've learned how drugs are discovered, developed, and brought to market. But one thing kept bothering us: what happens to the brilliant ideas that never get funded? In class and in journals, we've kept coming across research from independent scientists, some of whom had identified novel compounds, built models, or even completed small scale lab tests. And yet, because they weren't employed by a major pharma company or connected to institutional grants, their work stayed stuck in limbo. On solution does the mole address? First off, the mole unlocks liquidity for early stage drug discovery. Traditional pharma intellectual property takes years to get funding and to validate. Using Chainlink automation, Chainlink, CCIP, ELISA OS, and Avalanche Chain means all scoring, inference, and IP data are verifiable, tamper-resistant, auditable, and cross-chain. 3. Global collective research and development funding via DAO. Scientists, investors, DAOs, and patients can all participate in discovery and funding, not just big pharma anymore. I could come here, more my assistant, and say, I want to analyze a molecule. Or let's say I don't have any research and I want to use the most custom AI model that generates viable or eligible drug candidates, which require further research and development. I could use our AI by clicking molecule generation and get a nice little drug candidate there. I can take this and come in to analyze this molecule using ELISA OS. And if this molecule is considered found to be eligible or good, we are going to have a new token of the IP generated for this token. Let's connect. We are bringing real world assets. In this case, the asset is going to be our drugs or drug candidates, or in other words, smiles, to the blockchain. Okay. Now we've got our response from ELISA OS. All right. And what do we have here? I have analyzed the smile string that's given it to us. This molecule follows Limpinski's rule of 5 with a molecular weight of that. Okay, we have information about or description about this drug compound that we've been researching on. We have some more metadata about it, such as its toxicity score, its molecular weight, its QED score, its SA score. And then with that, we have that our token is considered a viable drug, meaning I can gather extra, I can put this for funding in the DAO. So this has been marked as eligible and transaction completed, and IP has been minted to the holding wallet. Use ELISA OS and our custom model as for inferencing and for scoring this drug molecule. If it is found to be a possible drug candidate, it is going to be queued on chain, and we will have Chainlink automation come in and pick it up and mint it for us. Now, this token can also be transferred to another chain, which is the Ethereum Sepolia chain, through Chainlink CCIP. Now, if we take a look at this token that was at the transaction ID, let's see if we can look at it on Etherscan. As our upkeep contract, we can see that our transaction has been queued. Our smile, our drug candidate has been our token itself. We can see a drug candidate has been minted for us. This drug candidate is being stored in a holding wallet where it is kept as our stake for providing this platform. Now, secondly, look at this. These are some of the existing uh, proposals that are going on. Users vote. and vote with collateral. And in return, they are given shares or intellectual property representing ownership of this drug candidate. Whatever revenues then made from research and production of this particular drug candidate is shared with all holders of the IP token for that drug candidate. We can come here, create an, a new proposal. Token ID for the last one we did was not two, was five. Paste in the name for it. Space that in there. This description is going to tell the other DAO members what this smile is, its potential, and what properties it has.